Of course.
Oh.
You have a father or grandfather who was a writer of textbooks of chemistry?
Yeah, my dad, Eugene Cordis.
Where did he teach?
At Indiana University, and then
I had a class with him.
Oh my God, that's awesome!
Biochemistry, I think. Had to be biochemistry. I had organic already.
Yeah, that's great.
Hello, everyone. Good
Fantastic.
evening.
I'll be mentioning it in my talk today.
Oh, I feel like I haven't been.
Another one of his books.
Good evening, everyone. Welcome to the Saddlebrook Science. Before we get started, I do just have a few quick announcements, so I'll make this short. For those of you who don't know me, my name is Samantha Pierce. I'm one of the program coordinators for the College of Science. Really fancy term for I get to come do fun things like this and facilitate talks. We are continuing on our series this month and next month with Mysteries Revealed, Discoveries in Chemistry. If you're new to the series, welcome. If you're returning, thank you. Just an FYI, for those of you who are interested or for those of you who have friends or family who weren't able to come tonight, the DesertView Performing Arts Center does record these talks. They do upload them to their YouTube page, as well as you can find them on our College of Science website. I upload all of those as well. And then we do have our YouTube, and then they definitely stash all previous series as well. And then that goes across the board for all of our outreach venues. For those of you who are new to the College of Science or new to the partnerships that we have across Tucson, we have 5 different outreach venues where we do host different science cafe talks, brewings. Hang on, I have my list here. The Downtown Science Cafe at Magpie's Pizza here at Saddlebrook, and then our newest one at the Tucson Botanical Gardens. And all of that information is listed in the brochure. If you go to our website, all of them are listed as well on the right side. There are a bunch of tabs, and then it gives more information regarding the presenters, what the series are about, different talk descriptions, that sort of thing. And sorry, I have my notes. And likewise up front, there's a bunch of brochures in the front regarding all of our different science outreach venues, Mirror Lab, Trainbreeding Lab, UA Sky Center, and then in addition, all of our upcoming events like Science City, which would be in March. So if you do have kids or grandkids coming in for their spring break, that's a really fun activity to take them to. And as just a reminder of our format, I'm going to introduce our presenter in a moment. He'll be presenting his research and what he has been accomplishing throughout our chemistry and biochemistry department here at the University of Arizona. From there, we'll do about 15 minutes of the roaming mics where we're going to do Q&A. As always, I'm going to give you the mic. You pose your question, hand it back to me so I can go ahead and run over to the next person and get their question on deck as well. And we'll do that for a period of time. Dr. Quartz does have a few kind of show and tell items here as well, and he'll be able to go into more details about that. And we do have some information regarding our chemistry and biochemistry department up front as well. Likewise, all of those links are on our website. And if you are interested in more specific information, definitely reach out to me. My contact information is as well on the brochure. And last tidbit before we get on to the good stuff. We do have the evaluations out front. I apologize. I think our student worker kind of got the documents confused. They are going to read Borderlands Brewing. It's meant for Saddlebrook, and our presenter is Dr. Matthew Cords. So ignore that part, but it's the same exact evaluation. That just helps us in better facilitating these talks, and then also it gives the presenter really good feedback in terms of, how we can improve this whole series for all of you. Likewise, there's a section at the bottom where you can add your email. We don't ever ask for donations. We don't spam you. I literally send out a once a month email regarding the different talks and just putting out an announcement regarding the upcoming series as well. And now let's get into the fun part. So I would like to introduce tonight's presentation, Venoms, the Chemistry of Life and Death, and presenting is Dr. Matt Cords. Dr. Cords graduated from Bowdoin College in 1987 with a Bachelor of Art degree in Chemistry and Romance Languages. He received his PhD in Organic Chemistry from Yale University in 1994 while working with Dr. Jerome Burson. And then he continued on his postdoctoral fellowship under Dr. Robert Sawyer at MIT until 2000. Dr. Quartz has been at the University of Arizona as an associate professor of biochemistry since 2001. He is fascinated by the evolution of biological molecules and focuses on the origins of protein folding and the evolution of venom proteins. And in his copious amounts of spare time, Dr. Quartz is a bluegrass and country music guitar player and singer. In addition, he does enjoy bird watching and hiking. Tonight, Dr. Quartz will discuss how the organisms involve venoms for the purpose of predation, and defense, sometimes by recurating proteins normally used for other purposes and modifying them for use as toxins. There's more, one sec. Also, he will discuss how components of venom can have therapeutic uses in human medicine despite the reputation of venoms for toxicity. Please welcome Dr.
Thank you, Samantha. The kind introduction. Venom, are you ready? Looks like a good audience. Tonight's a popular subject. Before I begin talking about this matter of life and death here, I want to actually start by mentioning and thanking some people. First, Samantha Pierce and Aaron Deely from the College of Science who've coordinated with me in putting this together, and everyone who puts together the Saddlebrook Science Cafe. Thank you for having me. I also want to say that some of the research that I will mention today was originally funded by a pilot project grant from the Bio5 Institute at the University of Arizona, which is actually funded by TRIF dollars from the state of Arizona. And that money allowed us actually to gain some preliminary results on this venom work that allowed us to put together a proposal for the National Science Foundation, which just got successful returning some research dollars to the state of Arizona. So I need to thank both the state of Arizona and the University of Arizona and Bio5 Institute, and then finally the National Science Foundation. As it says up here, I am a professor in the Department of Chemistry and Biochemistry at the University of Arizona. I also want to mention at the outset, I'd like to actually dedicate this talk to who is the founding director of the Viper Institute. And she worked tirelessly in the field of treatment of envenomation and antivenom, sane approaches to treating snake bites and other things. She was a tireless advocate for international cooperation in the subject of dealing with venomous animals and their bites, and she just retired in June 2008 with that work. I also need to thank at the outset, particularly this person here, Greta Binford, in the Department of Biology at Lewis and Clark College. She is the other half of the team that I'm part of. Greta is the biologist, the spider biologist, and I am the chemist. So we bring different expertise together to look at these problems of venoms and the molecules in venoms and their function for the organism. Greta is a fantastic scientist and has even been featured actually in a New Yorker article called and she's a terrific friend and collaborator. And I also want to mention that I won't be showing that much scientific data today, but some of what I'm going to show was done by a graduate student of mine, Dan LaJoy, who now lives in Boston. Okay, venom, right? So if you put venom into a search engine on the web these days, the first things that are going to come up are kind of popular culture items. You'll get the British death metal band Venom and their album Welcome to Hell. And more recently, you'll get the movie Venom, which is out in theaters right now, which is actually based on a Marvel Comics character, a rather villainous supervillain Marvel Comics character. Rating was a country at the box office for several weeks, which shows you that Venom sells. Right? It has a certain.
Just a little bit.
Okay, thank you. I'm getting used to the technology here. This thing is kind of hanging off my ear. Right. Is that a little better? Close. All right. I think my ear and my mouth are too far apart. Is that any better? All right. Okay, so Venom cells, popular culture, right? It has a certain gruesome, dark appeal in the popular imagination. But humor aside, if you went a little further down scroll a little further down in your search to look at what the substance venom means, what would you find? And you might find that it's time for a drink. And of course, if you used to go into a bar and people would say, "What's your poison?" This is a happy hour menu where one of the drinks is actually named the Rattler. So cocktails are sometimes compared to venom historically, and there is an apt comparison between cocktails and venom, not just because both are somewhat toxic, but also because venoms are mixtures of multiple ingredients generally. Okay, so if venom is a cocktail, what is it a cocktail of? Right, so you could ask, what's your poison? In the case of venom, the right question would be, what's your toxin? Because a toxin is a poison of biological origin, and that's what venoms have. Okay. So venoms are composed of a bunch of different kinds of molecules. Some of them are just little small carbon-based molecules. I just put the so-called molecular weight up here. That's about 320 hydrogen atoms versus of mass. That's a pretty small molecule. There are much larger molecules in venom called peptides. And this is one from a rattlesnake that's about 10 times the other. There are things called proteins and enzymes, which can be 10 or even like 100 times higher than that. And for example, some of the toxins in recluse spiders and some of the neurotoxins in black widow spiders are really rather large molecules. So we have very small, even though these are pictured at the same size, they go from very small to very large molecules. The other thing about venom molecules is that they have different functions. This one, tetrodotoxin, which is in octopus, the venom of some octopuses and other organisms, some worms, is a nerve toxin. This peptide toxin, crotamine from a tropical rattlesnake, is a myotoxin or a muscle toxin. And this we're going to talk some about this today. This Sphingomyelin Ace from a recluse spider is a necrotoxin, meaning that it causes tissue to die and decay. Okay. And there are also cytotoxins, which are things that kill cells in venom. So there are different classes of toxins of different size and shape. So a venom is a very diverse cocktail of molecules. So we're getting into the chemistry a little bit here. Mostly what I'll be talking about is peptides and proteins and enzymes. And in some sense, peptides, proteins, and enzymes are all the same kind of molecule. Right? All of those things are biological polymers that are constructed from building blocks in a chain. Okay. This is a venom peptide from a pit viper called tepratide. It has 9 amino acids strung in a chain, many of them the amino acid proline. Based on the order of those, we can define a sequence of amino acids for that peptide. A peptide is basically a chain with a small number of amino acids. If you get about more than 40 strung together, it gets big enough that it starts to be called a protein. But really a peptide and a protein are the same thing. It's just peptides are small and proteins are bigger, okay? And then a protein, the other term I want to define, a protein that carries out or catalyzes chemical reactions, as some of the proteins in venom do, is called an enzyme. Okay, so most enzymes are proteins. So peptides, proteins, and enzymes, all components of venom. Okay. I also want to mention that if you get bit by a rattlesnake, say, and you're rushed to the hospital, because these are complex cocktails, the primary treatment for these often is immunotherapy. Right, so antivenoms, I've got a couple of boxes of antivenom here. Antivenoms for things like snake bites will be generated by taking venom or venom components and injecting something like a horse with them, having the horse's immune system raise antibodies to the different venom components. And then when a human gets bit and they get injected with the antivenom, the antibodies from the horse will actually go and bind to and block these different components of the venom. That's the most widely used kind of therapy for venom bites. I'll actually I'm going to pass couple of these boxes around just so you can see this is a dose of green pit viper and cobra antivenom. You can open the box if you want, but there's 2 vials in it. One of them is just a liquid to dilute the antivenom, and the other is like a solid powder of the antivenom. Just don't open that vial if you look inside the box. Anyway, those venom, it's one interesting thing, and this has to do with Leslie Boyer's work, is that those antivenoms often only cost about $10, $20 to make a dose, but because of in the developing world, they might cost $50 or $100 on the market. In the United States, they cost about $2,000, $3,000 a dose, most of these. So if you get bit by a snake, you're in for a fair bill there. Okay, so it's also, I need to mention that proteins are things that are coded in your genes, okay? So you have DNA, which has a sequence of nucleotides, right? A gene will have a particular order of the letters of DNA in it, and there's a code by which that leads to instructions to generate the amino acid sequence of a protein, and that in turn encodes a 3-dimensional structure that protein has, and a function. In this case, this protein is binding to double helical DNA. So proteins are encoded by the information in your genes, and they can evolve as you acquire, as an organism acquires mutations. All right, now who has venom? What organisms have venom? The ones we know about most in Arizona would be things like reptiles. We've got a lot of different kinds of rattlesnake here. We've got Gila monsters. We've also got some venomous scorpions and a few venomous spiders. It is also true there are very venomous fish and rays, jellyfish, and it's not that widely known that there are some number of mammals that are venomous, including the duck-billed platypus, which has a little spur on its claw, for example, and many of the most ancient kinds of mammal species actually are venomous, and there's some thought that many more early mammals were venomous than there are venomous mammals now. There's even a venomous primate, and it's not a political figure in the United States. It's the slow loris. I thought of putting a picture in here, but I didn't. This is a very cute little primate, right? adorable. But it can lick this gland in sort of under its elbow, which has a bunch of venom in it, and then it can deliver a very nasty bite. It's also, its body patterning is also thought to be an evolutionary mimic of a cobra, which makes it it's both a venomous animal and it also, to some degree, to us it may look cute, but to many animals it may actually look like a cobra. That's an interesting venomous animal. Where does venom come from? And I don't really want you to look at this chart other than to say that these are all different kinds of protein that are present in venom. These are animals it's present in. And the main message of this slide is just that, venoms of things have a lot of different kinds of proteins in them. And what's happened is that the way proteins get into venom is that there's some kind of ordinary gene that encodes a protein. It gets duplicated and modified for use in venom, and then it gets selectively expressed or made in venom glands. Okay, so proteins thereby get recruited from other duties into use in venom. So this is quite common. And the same kind of protein, there's evidence, has been recruited many times in different venomous organisms, and this is known as convergent evolution, and there's a lot of convergence, in other words, coming to the same solution, different starting points. There's a lot of that in venom. Here's an example of it. Neurological toxins in venom act at these junctions between neurons and muscle cells, things like that, and affect the transmission of neurological signals in the system. And there are many ways you can do this, many things you can target to affect transmission of the neurological signals. And many different organisms have many of these different neurons and have recruited them independently into venom. Okay. Why have venom? Right. Well, the main reasons to have venom are either that you want to kill and eat something, your predator, like a spider, snake. Venoms are also used in defense, for example, in wasps and bee stings. There are some organisms like snakes that are very effective at using them both for predation and defense. Okay, so back to the movie, right? This movie, Venom, this is the poster for it. And it's going to be, the character Venom comes from an alien symbiote that occupies a human body, and the fusing of those two organisms leads to this supervillain Venom. But it has kind of a two-faced quality. There's the person and then the supervillain. And Venom has a little bit of a two-sided quality too, because it's composed of this cocktail of toxins which can harm and kill people, right? But as it turns out, there is another part to the story, which is that because there's so many biologically active things in venom, there's some potential also for some of them to be used as medicines. So I'm going to try to talk about both sides of that. I'm mostly kind of on the dark side, although you'll see that very early in my scientific research career, I was a little bit on the medicine side. We'll talk about that a little more. So I'm going to tell you two bedtime stories. And one of them, I just put two books up here just because to give you the idea that these are going to be stories. One is Richard Vedder. This is a very good book on the brown recluse spider. I'm going to talk about some research that Greta Binford and I have done on spiders related to the brown recluse and their venom. That's kind of the dark side, I suppose. I'm going to also some venoms in medicine. This is actually my father's book, Hallelujah Moments. He worked in drug development for many years, and this book actually contains one of the best stories of development of a drug from venom, and I'll be talking a little bit about that and other other venoms to drugs stories of recent import. Just begins with this family of spiders known as the Sicariids. It's a broad family of spiders. There's species in 2 genera or genus, Loxosceles and Sicarius. But one of the important things about this spider family is that it includes the notoriously toxic to humans brown recluse spider. Okay. Sicariid spiders in general have a pretty wide global range. They're mostly found in tropics or subtropics. Many in the United States, many in South America, many in Africa, some a little bit in China. One of the places they're not found, which is interesting, and it's just interesting because whenever you read about venom and toxic animals, venomous animals, people are always like, oh, Australia, they've got the worst animal, like the worst toxic animals, venomous animals in the world. But these spiders aren't in Australia, although this one from the Mediterranean has actually started to invade Australia a little bit. They have traveled there on planes or something. Okay, so first start by looking at one from Argentina, Sicarius, called the six-eyed sand spider. I'm just going to show you a little film. And basically all of these spiders are pretty venomous in the Sicariid family, but this one is kind of fun to watch. It lives, in the desert, in the sand. It's digging itself a little pit here, right? And you're going to see it do something that I like to do in a minute, which is to bury itself in the sand. So it's really not trying to just make the world go away, but it's actually trying to hide and ambush prey. So the relatives of these spiders, the Sicariids that are in the United States, they're not everywhere in the United States. There's a bunch of places that don't have any of them. These are all Loxosceles species, which are generally called recluse spiders. There's only one of those that is specifically the notorious brown recluse, and it's found in the Great Plains and the Southeast. But there are other species which are also pretty venomous that are found here along the southern border, including several species right here in Arizona to watch out for. There are parts of the United States that just don't have any of these at all, though. So sometimes people will report bites of recluses in Idaho or Washington or something like that, and that's not likely to be true. It's likely to be misidentified as something else. Okay, so this is a Loxosceles, a recluse spider. It's really big here, I might pass that they're going to be able to see it. These spiders are pretty small, even with their legs unfurled. They're only about the size of a quarter. People are like, he just brought a vial of a dead spider, where are the live rattlesnakes, But so it's a small, unassuming spider. It's got plain brown legs with thin hairs on it. It's got a pretty plain body. It does have this violin pattern on the back of the thorax. People sometimes call these violin spiders. But if you really want to identify one of these, it's not just the violin, it's these 3 pairs of eyes on the front. It's a six-eyed spider. That is the definitive thing. And I'm sorry about that. I hope that people are going to be okay with a few. If you come to a Venom talk, you're going to see a few gruesome pictures, right? So these recluse spiders tend to hide in dark places, under wood piles, closets, things like that. And people come into incidental contact with them. They're not aggressive toward people, but people can get inadvertently bit. And then after maybe a 24-hour period, you might notice this rather bad skin lesion, and it gets worse and worse. And finally, you may just have this sort of massive Necrotic Legion, meaning like a lot of death of the skin tissue. And those often have to be repaired with skin grafts. If you're really unlucky, and this is a 3-year-old girl from Tennessee who got bit on the chest here, and she's got a skin lesion developing, but this also can develop into a systemic syndrome where you get things like hemolysis, so damage to the blood, and ultimately, if you're, so you can get, this is a bloody urine sample, right? And ultimately, if you're really unlucky, you can die of kidney failure from this. That is not very common. There's only very occasional deaths in the United States. Deaths are more common in the tropics, places like Brazil. So this venom that these recluse spiders have is a complicated mixture, right? It's a cocktail. And you know, it causes all of these different biological effects that can produce the systemic and renal failure syndrome or the dermonecrotic lesions or both. If you're going to go after it, really maybe the best way is antivenom therapy, and there are antivenoms that have been developed in Brazil and Mexico. There's none available in the United States at this time. But the main thing I want you to look at here is there's one component of this venom called Sphingomyelin Ace. And Sphingomyelin Ace is the bad actor in this. It is primarily responsible for the worst effects here. And you can actually reproduce some of these effects just with the isolated Sphingomyelin Ace from the venom. And that was originally discovered by Brazilian scientist Denise Tamborghi, among other people. Okay, so I'm going to talk about Sphingomyelin Ace. What is a Sphingomyelin Ace, this bad actor in the venom? A molecule, a protein with the ending ACE is an enzyme. That's something that acts on something and catalyzes a chemical reaction. And a Sphingomyelin Ace is an enzyme that acts on the molecule sphingomyelin. Okay? And the molecule that gets acted on by an enzyme is known as its substrate. So this is a picture of the structure of the enzyme. It has a little here where the sphingomyelin molecule goes and gets acted on by the enzyme. What is sphingomyelin? Sphingomyelin is what we call a phospholipid. It has a phosphorus, and we're getting deep into the chemistry here. There's a phosphorus atom here, which is a part of what they call the head of the molecule, shown in red, which has these charged groups on it. It likes to be interacting with water. The other part of this, the lipid part of the phospholipid, are these long tails. They're greasy and they don't like to interact with water. When you put sphingomyelin in water, what it does is it organizes a bunch of sphingomyelin molecules in a row, and then it makes another, and it puts the two layers together so that all these tails are on the inside away from water where oil likes to be, and the head groups are up on the surface. And this can form a biological membrane that can be a barrier between the outside and the inside of a living cell. Okay. And sphingomyelin is a membrane phospholipid that makes up about 10 to 20% of your membrane, your lipid membrane composition. Okay. So how does my laboratory look at things like this? We want to study a toxin enzyme like Sphingomyelin Ace and what do we do? We use partly a technique called nuclear magnetic resonance. Right? This is a little bit like turning an MRI on its side, and instead of putting you in there, it's a little thin for that. You put a sample of molecules in there in this superconducting electromagnet, and you can see magnetic signals from the atomic nuclei. One of the things we like to look at in a phospholipid is the single phosphorus atom. Right? And what you can do with this is look at this signal over time. You take the sphingomyelin, you add some of the enzyme in that acts on it, and the sphingomyelin goes away. Its magnetic signal goes away, and something else grows in that's the product of the action of the enzyme on the sphingomyelin. So it's converting your membrane phospholipids into something else. The other thing that we sometimes do to look at enzymes is we grow crystals of them, shoot X-rays at them, get a diffraction pattern that we can then use to reconstruct the three-dimensional structure of the protein. Okay, so here's some things that we found by doing both of those. From the nuclear magnetic resonance experiment, we discovered for the first time that the reaction that this enzyme does on the sphingomyelin involves attack of an oxygen atom on this phosphorus with breakage of this chemical bond and release of most of the head group. And the product that leaves behind to be in the membrane, right, has this cyclic ring phosphate structure. Okay, I'll talk about why that's important in a minute. We also looked at the structure of the enzyme. That's what this sort of blob is. And from getting the structure, we were able to predict that it would interact with the surface of a membrane on a particular site of the enzyme. That's also the site of the enzyme we were able to predict was to be chewed up. And so what this enzyme is doing is it kind of runs along the surface of a membrane, and molecules of the lipid come up into the active site of the enzyme and get their heads chopped off. Okay, so this thing is kind of like a lawnmower that goes along the surface of a membrane and chops the top off of all the molecules. Now, lawnmowers, I don't know, some people think they're healthy for lawns, but this isn't healthy for your membrane lawn. And one thing about this is our experiments, or my student Dan LaJoie's experiment, showed for the first time that this molecule is the product. Nobody actually knows what this molecule does. It's actually a mysterious product, but because it's pretty different from the sphingomyelin, and in the head group, you might imagine that it modifies or damages the membrane surface. This is an experiment a different lab did where they took these so-called giant unilamellar vesicles, and that's a fancy term, but you can think of it as just like if you just made a plain, empty biological cell that was a sphere and just had a membrane and nothing inside it, really. And then to look at that, they put a fluorescent molecule on it so they could do fluorescence microscopy, treated it with this toxin from the spider venom, and showed that it really kind of messes up the surface of the unilamellar vesicle. So it's probably doing something like this to the surface of a cell. So messing up the cell membrane structure is one of the main things that this does, but it's more, there's more to it than that, because sphingomyelin has a particular role in cell membranes. And this is going to be from this movie, The Inner Life of a Cell. These blue things here, this is the surface of a membrane, and these are head group of membrane. And membranes, as it turns out, are not just these empty seas of lipids. They have all sorts of things floating around in them, like a fluid, proteins and other things. Sphingomyelin joins together with these sort of rigid regions of the membrane called rafts that move around in the membrane and carry important molecules with them. Okay, so this is just going to be a picture. This is a lipid raft made of sphingomyelin and cholesterol moving in the membrane. It's carrying these important proteins around on the surface of the membrane. So what we and many other scientists think is that, you know, when you hit this with the toxin and you modify the head groups of the sphingomyelin, it is going to damage these lipid rafts with some serious biological consequences. Okay, so you're going to get from the sphingomyelin some modification of the surface of cells and damage to the membrane. And we think that that's one of the main triggers that's leading downstream by complicated processes to dermatocrotic lesions and renal failures. Because if you modify the surface of membranes, one thing you can do, for example, is cause the immune system to react and attack the cell because it's been modified. So you can get all kinds of bad consequences leading to various kinds of damage and tissue death. Okay. Well, so this is what happens to humans and mammals when you hit them with recluse spider venom. But it's important to remember that these recluse spiders, their venom, these bites are very incidental. They don't show up for 24 hours. You know, if you're not really careful, you'd never associate the bite with a spider. The venom of a recluse spider probably didn't evolve to defend itself against, to defend the spider against us. The main reason the venom is there is to capture most prey, which are insects or other. And one interesting thing about that is that many of the prey of recluse spiders, like certain flies, actually don't have any sphingomyelin, even though the spider has all these sphingomyelin aces in the venom. And it turns out, in fact, that flies have, instead of sphingomyelin, they have a different phospholipid called ceramide phosphoethanolamine. Other arthropod prey items have both ceramide phosphoethanolamine and sphingomyelin, whereas vertebrates, like people, really primarily just have sphingomyelin. But we looked at this and we found that actually some of the toxins in recluse spider venom act preferentially on sphingomyelin, some of them act preferentially on ceramide phosphoethanolamine, and some of them have relatively little preference. And so we think that recluse spider venoms may have evolved to specialize possibly for the lipids present in some different types of prey. So there's some significance of this for predation by the spider. And it turns out, specifically on ceramide phosphoethanolamine, as you might expect, are actually less toxic to vertebrates and people. So only some of these enzymes in the venom actually are what caused people problems. Okay, so what actually do these toxins do to the prey? Insects also don't have skin to decay, right? So are these necrotoxins to the prey? Well, they're probably not. There's actually evidence from my collaborator at Binford's lab that they're neurotoxins, and this poor creature is a cricket. It's a little tough for me to watch, actually, but this is a cricket that has been exposed to purified toxin. And you're going to see that it is moving its legs in a very funny and unusual way here. It's actually having difficulty moving properly. It's going to go on for a little bit, flailing about a bit, extending the legs, twitching the legs. And I think I will actually skip ahead and just show you, to spare you the rest of this. After about a minute, the cricket's completely paralyzed. So these things to prey are actually probably neurotoxins, right? We don't really understand yet the detailed action behind this that makes them nerve toxins, but that's probably what they're being used by the recluse spider for in prey. I'll just mention briefly that these toxins come from somewhere. Remember that proteins get recruited into venom from other sources. These are related to a very ordinary, what we'd call a housekeeping protein that every organism has, but it's been modified in several ways to be able to act on lipids in the biological membranes. Okay. But even this, this family with these features actually of Sphingomyelin aces has actually been around for hundreds of millions of years. And we know that actually because it is conserved in all arachnids, although it is only in the venom of certain arachnids like recluse spiders, scorpions, and ticks. And it appears to actually be present. We don't know what it normally does in arachnids, but it has been recruited into venom at least 3 separate times. Versions of these enzymes are also present in things like comb jellies, which, like spiders, are clever predators. Some corals actually utilize these, and some pathogenic fungi and bacteria as well. In fact, versions of these toxins have been found in the valley fever fungus, for example. Okay, so I'm going to spend, I may have to go over a few minutes. I'm going to spend the last little bit of time here talking about the other side of venom, that of venoms in medicine, right? And I will say at this point that, you know, again, I said I'm maybe more on the dark side of the venom picture, looking at their toxic activities, role in predation and things like that. And I've only actually been working on that for about less than 10 years of my career. I only came to study venom. I was sort of a more general protein evolution scientist, and I only came to study venom proteins by meeting Greta Binford, and you know, she came to me and said, I've got this really interesting protein in recluse spider venom that I want to look at, and I need somebody who knows about proteins to look at this. And that's how we started the collaboration. But most of my scientific career before that, I didn't work on venom at all. But after I started working on venom, I actually realized that my very first research experience, actually, when I was about 16 or 17 years old, had a lot to do actually with a classic case of venom being developed into medicines. Now I realized I had sort of come full circle. So that's this last little bit of the story about developing drugs and developing them from venom. So my father had said one summer when I was a teenager, he said, basically, he's like, you need something to do in the summer. You know, he didn't, I think what he said, he didn't want me just hanging out, right, downtown or something. So he said, why don't you come in and work in the lab in my company, which was Merck. And he put me to work with these wonderful people, Herb Bull and Nancy Thornberry, who are just the greatest folks in the world. And they gave me a wonderful early impression of doing research science. And I wound up with my first research publication from those first couple of summers of work at Merck when I was about 17 years old. It has this name, and it had to do with this thing called angiotensin converting enzyme and its inhibition by these molecules with complicated names. And it turns out, so angiotensin converting enzyme is a enzyme that acts in a complicated hormone network. And you don't need to understand anything about this slide, except that this little hormone here, angiotensin 2, one of the things that it does is to control or increase blood pressure. So if you're looking for a blood pressure drug, one strategy was to try to block the formation of angiotensin 2. And the way to do that was to block the enzyme that converted this angiotensin 1 into angiotensin 2. Okay, that is called angiotensin converting enzyme, or ACE. And it's potentially a target for blocking the formation of this and lowering blood pressure. And some scientists scientist named Ferreira, I believe, was looking at venoms from tropical pit vipers and pulled out some peptide things that turned out to have activity as inhibitors of angiotensin converting enzyme. Okay, and this was great in one way and not so great in another. The problem with them is that you couldn't take them orally like a pill. They just kind of go through the system or get chewed up instead of being absorbed. So to take them as drugs, you'd have to inject them, and nobody wants to inject something for a high blood pressure drug, right? So they were not orally bioavailable. But the injection studies did succeed in showing that using these angiotensin converting enzyme inhibitors was an effective therapy and a safe therapy against high blood pressure, right? And then scientists at two different companies took the peptides that Ferreira and others had discovered and modified them to keep some of the important parts. And one of those drugs, and make orally bioavailable drugs, and one of those drugs became Captopril from squib, which was a major early high blood pressure drug. And then it had some safety problems, and scientists from Merck, including my father and Art Patchett, worked on developing it into a better and safer drug, and came up with this. But you can see there's some resemblance here between that and the original snake venom peptide, and that actually became the most successful of all of the angiotensin converting inhibitor drugs for treating high blood pressure. But that was venom inspired, right? It started with venom and then led to a venom derived drug. And it turned out that, unbeknownst to myself, I worked on this as a teenager and I never really understood the history of it. You know, I was, okay, I'm looking at these molecules and this enzyme, but it never really dawned on me at the time that they had actually been derived from snake venom. So after I started working on venom in recent years, I looked back at that and thought, hey, I kind of came full circle. So there are these other venoms, other venom molecules from different organisms that have been developed into marketed drugs. There haven't been a huge number of them. You know, only a half a dozen over the years, but many more are in development. And they come, for the most part, from rattlesnakes and other kinds of viper. And many of these, because rattlesnakes have a lot of things that act on hemostasis in the circulatory system, some of these either cause or prevent blood coagulation. So some of these anticoagulation things are used to prevent heart attacks. There are diabetes treatments that come from the Gila monster, pain treatments that come from the cone snail, and of course the original Captopril drug for high blood pressure. One of the most interesting recent examples of venoms to medicine is something called Tumor Paint. And scientists working on venom of the Israeli deathstalker scorpion discovered that there was a peptide in this venom that, for some reason that I believe is still not that well understood, binds really selectively to cancer cells, okay? So one of the things that they thought of to do was to fuse the toxin to a fluorescent molecule and put it in a cancer patient and use it to image tumors. And one of the really good uses of this potentially is that some things like certain brain tumors, it is really difficult to define the boundaries and the exact location of the tumor cells. So if you can light it up using these imaging agents, it's a major potential advance for brain surgery for successfully removing the entire tumor without damaging other tissue, especially in the brain. So that's a really interesting thing. I believe this is not FDA approved yet, but it's in late phase clinical trials. Okay, so that's another interesting example of venoms to medicine. I will just leave you with this one. Natural Viagra. It's from a very reliable news source. But this is a real thing. This is, I think, a South American spider, rather much scarier looking spider than a brown recluse. It's rather large. So, you know, I don't think it's worth it, probably. But anyway, that is all. I thank you for coming out tonight and hearing.
Thank you, Dr. Cords. Do we have any questions? We'll do roaming mics for about 15 minutes or so.
What were the name of the two venomous mammals besides the platypus? Yeah.
The question was, what is the name of the two venomous animals? One of them is the Sloloris, which is the which is the primate, right? And then the one on the left in that image, I'd have to go all the way back, but I'm actually not sure. We go all the way back to the beginning. Oh, it's a shrew of some kind, I think. Yeah, but I'm not sure. Yeah.
But some animals seem to have an immunity to venom, like, for instance, rattlesnakes biting bull snakes who seem immune to that. Is there an explanation for that?
You can gain well, so there's some kind of natural immunity, right? Humans don't generally have natural immunity to most venom, which is why they have to be treated with antivenom that's derived from injecting a horse with the toxin. You can actually, so that's taking an immunized horse and then taking the antibodies that come from that, and when you inject in the human, it doesn't immunize the human, the antibodies just block the toxin. But you can immunize a human against many venoms. I think some snake handlers have successfully immunized themselves by exposing themselves to small amounts of the snake toxin. So you can have acquired immunity. But in terms of animals that have, that have natural immunity against venom, that's not something I know a lot about, actually. So I don't think I could address that. But certainly humans can acquire it. But in cases where some animal has natural immunity to a cobra venom, I'm not sure how that works.
Do same species, are they all immune to each other's venom? Or are they immune themselves to their own venom?
Are they immune to their own venom? Yeah. There's interesting cases where, the question, I'm not sure about the answer to that specific question. Are animals in the same species immune to their own venom or other members of that species? I do know that issues come up like, there was an interesting paper a couple of years ago on why, poison dart frogs that you find in the tropics that have these awful neurotoxins on their skin and stuff, why they don't get poisoned, because they actually have basically the same neurological protein receptors that people do that get attacked by the neurotoxin. The question was, why don't the frogs end up killing themselves with their own toxin? And the answer was that they figured out was that they had subtly mutated these receptors so that they would still be able to function, but would no longer be susceptible to the toxin. So that sometimes does happen, that animals will actually evolve defenses against their own venom toxins.
The Sonoran toad, is that an actual venom from the Sonoran toad in the parotid salivary gland?
I do not know. I'm just going to have to say I don't know that one. Yeah, the question was with the Sonoran toad. Can you refer to its saliva as a venom? I'm not sure. Yeah,
And
so I'm
then,
not sure what it
and then how
contains.
toxic are those bark scorpions that seem to be here?
Pretty toxic. There's a funny thing with scorpions that's known to be the case, that the size of the pinchers is inversely related to how venomous, how harmful their venom is. And I think it's thought that a scorpion with larger pinchers is able to subdue a potential prey for longer, to be able to sting it more time maybe, whereas the ones with the small pinchers need to be able to kill something faster. But those two things are known to be oppositely related that way.
And how do they treat, how do you treat for the bark scorpion?
I don't know of a, maybe someone else in the audience knows this, but I don't know of a treatment for it. I mean, I know that sting is extremely painful. I don't remember if it's ever life-threatening to children, for example, or if you get many stings, if it's life-threatening. But I don't know of a treatment for it.
Is it a neurotoxin? Do you know?
Scorpions certainly have a lot of neurotoxins in them. Yeah.
I have a question here. How long does it take for the toxins to work in your system? How quickly do they go in?
You mean in say a brown recluse spider? Yeah. That it's not fast. It takes, in a recluse spider, it often takes, there's some cases where a systemic syndrome has actually been known to be able to kill a person within a day or two of a bite. The Necrotic lesions usually don't appear at all for at least about a day, and then it takes a while for them to grow really bad. So it's a fairly slow acting toxin.
When a human is injected with venom, the venom must metabolize or something, must degrade. Has anything been considered that you could cause the venom to metabolize and become less toxic to humans?
Cause the venom, it's the question that, like, as a treatment, could you cause the venom
Treatment that could metabolize.
to degrade or break down? Yeah, I don't know. I don't know the answer to that. I do.
Essentially what the immunotherapy is going to do.
Yeah.
It's going to bind the toxin, and then that's going to allow it to be processed by the cellular immune system. So it's kind of the same thing. Not exactly.
Right, and
Exactly the question, but
Mhm.
it's kind of, that's what they hindered the antibody. The complex. It would be inert.
yeah,
Kind of the same.
yeah, that's a good point. And I was going to mention that, yeah, along those general lines, the general strategy of administering antivenom is generally has been the best way of going after the venom directly and binding to it and stopping its action.
If you get bit by a scorpion or a spider, is there a threshold of amount of toxin that you have to have before you see a serious result? In other words, is it dose sensitive?
Is there a threshold of toxin or venom that makes it medically serious? Is that the question? Yeah. It's certainly true that it's certainly, I think, known that say not all, it's actually only a relatively small percentage of recluse spider bites that lead to dermatocrotic lesions, for example. Maybe somewhere around 10 to 20% of bites actually lead. I'm not sure if it's known why that is, but it's quite possible that, it has to do something to do with degree of envenomation. And of course, rattlesnake bites vary quite widely in how much venom is delivered, and that can have a big relation to the seriousness of the bite.
So a short story. My father was bit by a brown recluse. The last night we were in Texas heading up to Maryland. And during the course of that drive, it went from just being like a little tiny bite to an open sore about the size of a quarter. Fortunately, when he checked into a hospital up there, the doctor who saw him had just come from Texas and recognized it. And in the end, he had to have that whole area around his ribs excised to be able to get it to finally stop.
Yeah, that's, of course, Texas is definitely a place where you will find members of this spider family, and that is a story that with the bites that does often happen. With, I don't know how long that drive took, but yeah, over the course. Yeah, that sounds about right. Several days to a week, and then they can spread so bad that really the thing to do is just to cut the tissue out around it. Yeah.
I was stung by a bark scorpion on the tip of my finger, and it was late at night, and I treated it like a bee sting. I took some Benadryl and then put ice on it.
Mm-hmm.
And it did sting quite heavily. And after about an hour or so, the pain went down. It still was there, but I was able to sleep. And then afterwards, that finger became numb for like a week.
Mm-hmm.
And there was no feeling in it, but there was no discoloration. There was no after effect or anything to it.
Yeah, so you just got mostly a localized neurotoxic effect. Yeah.
We'll go ahead and do one more question if there is one. All right. Well, thank you again so much, Dr.
Thank you
Cords.
so much for listening.
And if I could bug whoever has the pass around products, the anti-venom, to please either find me or take them up to Dr. Cords. And then just an FYI, the next Science Cafe will be Tuesday, December 11th. So Tuesday of next month, December 11th. And then our evaluations are up front. Thank you so much, everyone. Have a great evening.